Okay, well, these videos keep cutting me off, and it's going to get real annoying, but y'all are going to have to bear with me. Anyways, like I was saying, when you do ratios, you need to pay attention that every time you're having a genotype ratio, you always end up with 3 numbers down here. Now, you have to understand it is possible that if I were to do true breeding, for example, and I took 2 completely dominant purple flowers and crossed them with each other, all of the offspring would also have big P, big P. I would have no offspring that had big P, little p, and no offspring that had little p, which is fine. So when I did this ratio, I would have to say that ratio would be 4 to 0 to 0. Those zeros are important. A lot of people don't want to write them because they think they're insignificant, but they are very, very important. If I just put a ratio of 4, that's not really a ratio at all. So every time you do genotype, you should have 3 numbers in your ratio, and every time you do a phenotype, I should have 2 in my ratio. So if I had 100% purple flowers, for example, that would be acceptable. I would just have to say that the ratio is 4 to 0. Okay. These are some more words that are very important that you understand. Homologous and heterozygous. Y'all should already know what homologous means. It means having the same. So when I talk about homologous chromosomes or homologous alleles, I'm talking about that they're the same. Heterozygous means that they're different. So if I have, for example, 2 big P's, those would be considered homozygous dominant because they both have the same dominant gene, and if I had 2 flowers that had a big P and a little p, they would be classified as heterozygous because their alleles are different. And if I had 2 little p's, they would be classified as homozygous recessive because they have the same recessive allele. Okay, so as you remember, my ratio for genotype is 1 homozygous dominant, 2 heterozygous, and 1 homozygous recessive. And my phenotype would be a ratio of 3 to 1 of 3 purples and 1 white. So even though these are homozygous dominant and heterozygous recessive, heterozygous, they all show up as purple. So I clump them together in the same grouping when I'm talking about phenotype. So one of the problems that Mendel came upon in his experiments is that he knew that you could have 2 purple flowers and one could be considered true bred or it could be homozygous dominant, meaning both of the alleles were dominant. Or you could also have a purple flower that looks the exact same and it would be classified as heterozygous, meaning it has that secret white allele hidden in it. But it's impossible to tell just by looking at the flower. So he developed a way to determine how you could figure out if you had just a homozygous dominant purple flower or if you had a heterozygous purple flower. So the way that you would do that is you would take your unknown flower and you would cross it with something you did know. So I know that if a flower is white, it has to be homozygous recessive. There's no other option. It has to be little p, little p. So if I can cross this unknown purple flower with a homozygous recessive flower, I should be able to figure out if this flower is big P, or big P, little p. So basically all you have to do is I would cross my test flower with my homozygous recessive flower. If all of the offspring came out to be purple, then I know that my mystery flower had to have been a homozygous dominant purple flower. But if I had offspring where half of them were purple and half of them are white, that tells me that my mystery flower had to have been heterozygous. So I can figure out by looking at the offspring and track backwards and figure out what the parents' alleles were. Okay, so we need to talk a little bit about Mendels Laws. So his first law is the law of segregation, which basically means that each allele for a trait is packed into a separate gamete. Okay, so if I had 2 alleles, a big P and a big P, one of my big P's would go to one gamete, one of my big P's would go to the opposite gamete. Same thing for here, I'd have one little P go to one gamete, one little P go to a different gamete. So if I was heterozygous, I would have one gamete that had my dominant allele and one gamete that would have my recessive allele. So as you remember, you start off with 2 homologous chromosomes. those are going to divide and then divide again so that I can have my 4 gametes. So I will have only one side of each chromosome in each one of my gametes. So I'll make, let's say that this this is purple and this is white, so I would make 2 gametes that had the purple gene and 2 gametes that had the white gene. This law of segregation takes place in meiosis 1 at metaphase 1. So when my chromosomes line up and they do independent assortment and they do crossing over, that's what helps me segregate my genes so that I'm only passing on one down into each one of my gametes. So far we've only talked about Mendels monohybrid cross experiments where he took purple flowers and crossed them with white flowers. When I do a simple Punnett squared, that's called a monohybrid cross. But if I were to cross more than one characteristic, then that would be considered a dihybrid cross. So Mendel also did experiments with peas where he crossed yellow and green peas that were both round and wrinkly. So he has 2 different alleles working at the same time, seed color and seed shape. So he's trying to find out how many round green peas there are, how many yellow round peas there are, how many yellow wrinkly peas there are, and how many green wrinkly peas there are. So there's many different genes going on at the same time, and he's trying to figure out how you would cross all of them at once. So in his basic experiment, what he did is he took one yellow round pea and one wrinkly green pea. He crossed it and he found out that the first generation were all yellow and round. And then he took this generation, self-pollinated with itself to make a second generation. He found out that 9 out of 16 of them were yellow and round, 3 out of 16 of them were green and round, 3 out of 16 of them were yellow and wrinkled, and 1 out of 16 were green and wrinkled. So it gave a ratio of 9, 3, 3, 1, and that would be for the phenotype. So Mendel knew that he had 2 different genes working at the same time. So let's just say we had a big Y and a little y and a big R and a little r. These are what the alleles were for that first generation. So I had a dominant allele and a recessive allele, a dominant and a recessive that came from the yellow round parent and the green wrinkly parent. And he was trying to figure out if they made gametes this way or this way. So he said you could have either taken the big Y and the big R and made one gamete, and the little y and the little r and made one gamete. Or was it possible that you had 4 different gametes of several different variations? So he took the big Y and the big R, that's one. The big Y and the little r, that's another variation. The little y and the big R is one, and the little y and the little r is one. So after looking at it, he discovered the only possible scenario to get all of the findings that he saw in his data is if the genes actually had this completely different crossing over variation happening where he had 4 completely different gametes. So what you would do if you're doing a dihybrid cross, you would write the 4 possible gametes of one plant, the 4 possible gametes of the other plant, and you would do this giant 16 square dihybrid cross where I would have 9 out of 16 would be yellow and round, 3 out of 16 would be green and round, 3 out of 16 would be yellow and wrinkled, and 1 out of 16 would be green and wrinkled. These dihybrid crosses are something that can be very tricky to do, and they're very easy to mess up on. So whenever you start doing them on your own, make sure that the very first thing you do is you take whatever alleles you're given for that plant and you separate them into the 4 completely different possible gametes. And each one of those gametes will go up here either along the side or along the top of the side, and you'll just fill in the table accordingly. And you're going to have to be very, very careful when you write your letters and very careful when you count to make sure that you're doing everything. There is one giant hint though. This 9:3:3:1 ratio is a very, very important ratio. Anytime that I take 2 completely heterozygous alleles for both the color and the shape, two completely heterozygous for both parents, the ratio will always be 9:3:1. So I could do this for hair color and hair type, but if the parents are both heterozygous for both of them, I know that their ratio is going to be a 9:3:1 ratio. It doesn't matter what two alleles I'm crossing. If both of the parents are heterozygous for both things, it's going to be 9:3:1. And that's a number that you should memorize because it'll save you a lot of time. That's the ratio that shows up most of the time is a 9:3:1 ratio for tests and stuff. So instead of doing this whole big chart, it'd be easier if you just recognize, oh, here's heterozygous, this is another heterozygous, that's heterozygous, that's heterozygous, oh, they're heterozygous for everything, so I can just say they're going to be 9:3:1. So y'all already know about independent assortment, but after Mendel looked at the possibilities of how the gametes were made, he realized that you didn't just always have the same two pairing up together. You had independent assortment, which means it's a random variation. So I could have had a big Y and a big R, a big Y and a little Y and a big R, or a little Y and a little r. So there's four different possible combinations. And the only way that those could line up that way is if independent assortment happened during meiosis. So they line up in a random variated way, and that's how you get those four completely different gametes. So this law of independent assortment is a pretty big deal, and I know that y'all remember it happens in metaphase 1 of meiosis 1. But what's very important about this is that Mendel didn't even know about DNA or genes. They weren't even discovered yet. So he was already identifying different, I don't know what to call them, scenarios that were going on during meiosis without even knowing what meiosis was or what DNA was. So he was really ahead of his time, and he helped lead the way to making other connections to DNA and all the rest of the stuff. So he's a really important guy. So when they first started studying meiosis and DNA and chromosomal replication, a lot of people took the studies of Mendel and they were able to basically prove their theories based off of Mendels experiments by using these different chromosome pictures right here where you show how meiosis happens, which I know y'all know this, but they were able to apply the principle of segregation and the principle of independent assortment to help prove that all of the gametes at the end are not going to be the same. They're all going to be different variations. So Mendel really helped pave the way for all of this. Okay, so just to review, Mendel's two very important laws are the law of segregation, which happens when you do monohybrid crosses and you're just dealing with one single trait. It says that each allele separates into separate gametes. And then you also need to know about the law of independent assortment, which happens when you're dealing with more than one trait, so you're doing a dihybrid cross. And that means the genes on separate chromosomes assort into gametes independently. Both of these things happen in metaphase 1. There is exceptions if you have linked genes, but we're going to talk about those later. So there are certain genes that are so closely related that they always stick together. But you're not going to learn about that until a little bit later.